Always a protein molecule starts or begins with an amino acid called methionine. Primary structure is found in the form of linear sequence. Hemoglobin is a protein which is a carrier of oxygen. So what is a phosphodiester bond? The bond between the phosphate and the hydroxyl group of sugar is an ester bond. So enzymes isolated from thermophilic organisms live under high temperature. They are called as thermostable. Hello everyone, welcome back to session 5 of chapter 9 called Biomolecules. In the last session, we have studied about a very important biomolecule, that is carbohydrates. So as we already studied, the very important biomolecules are carbohydrates, proteins and lipids, and the micromolecules are the enzymes and vitamins. We all know how important these biomolecules are for the body. And as we know, the building blocks of carbohydrates are monosaccharides, and the building blocks of protein are amino acids, and the building blocks of lipids is fatty acid and glycerol. So now let us study about the structure of proteins. As we all know, there are 20 different types of proteins that make a protein. There are 20 different types of amino acids which make different proteins in our body. There are many more amino acids found in the body, but the very important 20 amino acids are required to make a particular protein. And I think all of us know the protein, the sequence of amino acid for a particular protein varies from a protein to a protein. For example, hair is a protein, nail is a protein, eye lens is a protein. So each protein has a particular structure, and that sequence depends upon the amino acid sequence, as we already studied in the previous session. So the amino acids join like this, and always the beginning of the protein chain starts with methionine. We have different types of amino acids like alanine, cytosine, thymine, guanine, right? So like that we have 20 different types of amino acids, but one such very important amino acid is called methionine, which is always called as a starter. So for anything there is a beginning as well as an end. So always a protein molecule starts or begins with an amino acid called methionine, and I think all of us know that each amino acid is coded in the form of triplet codon system. For example, methionine is coded as AUG, right? AUG is the triplet codon system, because as we know, there are 20 different amino acids, and singlet codon system, doublet codon system will not satisfy coding for 20 amino acids. So the triplet codon system, that is, what is triplet codon system? Triplet codon system. It is the system for coding, that is usage of 3 nitrogenous bases coding for a particular amino acid. So each amino acid has a particular triplet codon. So that codon will help in coding for a particular amino acid. For example, like methionine is coded with a triplet codon called AUG. So for any protein molecule, it will begin with the methionine. So it is called as a starter codon. So any protein molecule, may it be a nail or a hair, so any protein will start with the methionine. So it is called as starter codon. So in a similar way, there is an end for a process of protein synthesis, and there are terminator codons. What do you mean by terminator? Stop codons. So once the codons will come and sit here, they are called as stop codons. So there are 3 stop codons, right? So they will come and sit here. So when they come and arrange here, that means the process of protein synthesis will stop, and the whole molecule twists like this, and that is how a protein molecule is formed. So this whole process, we call it as what? Protein synthesis. We call it as protein We call it as protein synthesis. So now let us study the structure of proteins. Here you can see the structure of proteins. So we can study the structure of proteins as two very important structure. One is called the primary structure, the other one is called the secondary structure. And as we all know that the amino acids are held by a peptide bond. What is the bond between two monosaccharide units? In case of carbohydrates, the two monosaccharide units are joined by glycosidic bond. But whereas in case of protein, the two amino acids are held together by a very important bond called peptide bond. So now let us study the structure of a protein. So it is found in the form of linear sequence of amino acids. So this is called as a primary structure. First amino acid is called terminal amino acid and last amino acid is called, the first amino acid is called starter amino acid. And the last amino acid is called terminal amino acid. Terminal means terminate, means with stop the process. So primary structure is found in the form of linear sequence. So it's like a straight structure. First amino acid is called as what? As I said, it is called as starter amino acid and last amino acid is called terminal amino acid. Moving on to secondary structure, polypeptide chain undergoes folding. Here you can see the folding and coiling. So this is the secondary structure. This is the secondary structure of protein. Secondary structure of protein. So polypeptide chain undergoes folding or coiling, which is stabilized by hydrogen bonding. Right-handed helices are observed. Example like fibrous protein in hair and nails. So what are the different types of structure of proteins? We have the two very important structure called primary structure and secondary structure. So it is found in the form of linear sequence of amino acids. So first amino acid is called as what? Starter. So usually the starter will be what? Methionine. An amino acid called methionine. And last amino acid is called terminal amino acid, which is otherwise called as, they are also called as nonsense codons. Nonsense codons. What do you mean by nonsense codons? Codons which makes no sense is called as nonsense codons. So codons which is not going to code for any amino acid. As I said, each amino acid is coded by a particular triplet codon system. That is what is like, for example, methionine is coded by AUG. So this is called as a triplet codon system. So this is called as triplet codon system. What is triplet codon system? Usage of three nitrogenous bases coding for a particular amino acid. So what is secondary structure? Polypeptide chain undergoes folding. So this is called as what? Secondary structure. So the one which is in the form of a linear sequence, we call it as primary structure. And the one which is folding or coiling, which is stabilized by hydrogen bonding, right-handed helices are observed. So you can see the right-handed helices. Helices means twist. Here you can see the right-handed helices. So for example, like we have the fibrous protein in hair and nail. That is about the structure of proteins. So tertiary structure. So what are the four types? Primary structure, secondary structure, 3 tertiary structure, then 4th quaternary structure. So here you can see the multiple quaternary structure. Here you can see the tertiary structure. So the previous diagram was, here you can see this was secondary structure. And if the amino acids, they join like this, this is called primary. This is called the primary structure. This is called as the primary structure, right? So this is called quaternary structure and this is called the tertiary structure. So what is this tertiary structure? Long protein chain is folded upon itself like a hollow woolen ball. Here you can see it is like a hollow woolen ball. So it looks like a woolen ball folded upon, like if you take woolen and if you start folding one above the other, then it becomes like a ball-like structure. So it looks almost similar to that. Hence, that type of structure of protein is called tertiary structure. So long protein chain is folded upon itself like a hollow woolen ball. Gives three-dimensional view of protein. Example, myosin. Actin and myosin are what? Contractile proteins. So this myosin is called as a contractile protein. Contractile protein. So that which helps in contractility. I think the movement is made possible with the help of muscle proteins, namely actin and myosin. So myosin, which is a muscular protein, is nothing but a tertiary structure. Then moving on to the fourth one called quaternary structure. Two or more polypeptides with their folding and coiling are arranged with respect to each other. So here you can see how they are arranged with respect to each other. So like human hemoglobin. I think all of us know human hemoglobin. Hemoglobin is a protein which is a carrier of oxygen. So molecule has what? 4 peptide chains, 2A and 2B subunits. So what is tertiary structure? Long protein chain is folded upon itself like a hollow woolen ball. Gives three-dimensional view of protein. Example you can give is myosin. Then quaternary structure, 2 or more polypeptides with their folding and coiling are arranged with respect to each other. Example, human hemoglobin molecule has 4 peptide chains, 2A and 2B subunits. So this is the tertiary structure. And this is the quaternary structure. So what are the 4 types of protein structure? Primary, secondary, tertiary and quaternary structure. The nature of bond linking monomers in a polymer. So here you can see the bond between 2 amino acids. So this is one amino acid and this is another amino acid. This is another amino acid and this is another amino acid. So this bonding is called as the peptide bond. Just like 2 monosaccharide units are held together by, in carbohydrates, it is glycosidic bond. And in proteins, it is peptide bond. So here you can see that between, as you know, protein molecule has an amino group at one end and a carboxyl group at one end. Amino group at one end and COOH group at the other end. Right. NH2, that is NH2 group. So nature of bond linking monomers in a polymer. Peptide bond formed between the carboxyl group of one amino acid and the amino group of the next amino acid with the elimination of water moiety is called the peptide bond. So what is a peptide bond? Formed between carboxyl group of one amino acid and the amino group of the next amino acid. Here you can see the carboxyl group of one amino acid with the elimination of water moiety is called as a peptide bond. Here you can see the peptide bond. So the 2 amino acids are held together by the peptide bond. So peptide, what is a peptide bond? Formed between the carboxyl group of one amino acid and the amino group of the next amino acid with the elimination of water moiety. Next, glycosidic bond. As I said, individual monosaccharides linked with each other to form polysaccharides. Then this bond is also formed by dehydration, formed between 2 carbon atoms of 2 adjacent monosaccharides. So what is the nature of bond linkage, linking monomers in a polymer? Peptide bond, you know, this chain is called as what? Polypeptide chain. Polypeptide chain. What do you mean by poly? Many amino acids many amino acids are linked with the peptide bond. So formed between the carboxyl group of one amino acid and the amino group of the next amino acid with the elimination of water moiety. So glycosidic bond, individual monosaccharides linked with each other to form polysaccharide. So this bond is also formed by dehydration, formed between two carbon atoms of two adjacent monosaccharides. Then phosphodiester bond. So it is a nucleic acid, a phosphate moiety links the 3 carbon of one sugar, one nucleotide to the 5' carbon of the sugar of the succeeding nucleotide. Nucleic acid, what are the 3 very important components? What are the 2 types of nucleic acids? That is DNA and RNA. So what are the chemical components of DNA then? That is, it is made up of a sugar called deoxyribose sugar. Then it is made up of 4 nitrogenous bases. In case of DNA, it is AGCT. Then it is made up of phosphoric acid. So the backbone of the DNA molecule will be what? Sugar plus phosphoric acid. So as we know, it looks like a ladder. So sugar, phosphoric, sugar, phosphoric, sugar, phosphoric, sugar, phosphoric. So here A with T, C with G, G with C, T with A with T. So this is called nitrogenous base pairing. As we know, there are 4 nitrogenous bases. 2 are purines and 2 are pyrimidines. Purines will never base pair with purines nor pyrimidines with pyrimidines because of their stability, as we already know. So the bond between, so in a nucleic acid, a phosphate moiety links the 3' carbon of one sugar, one nucleotide to the 5' carbon of the sugar of the succeeding nucleotide. So one will be 5', one will be 3'. So this will be 3', this will be 5'. So what is the bond that is formed? It is called as phosphodiester bond. So what is a phosphodiester bond? The bond between the phosphate and the hydroxyl group of sugar is an ester bond. So there is one such ester bond on either side. It is called as phosphodiester bond. So there is one such ester bond on either side and it is called as phosphodiester bond. Moving on to the 2 very important types of pathways called anabolic pathway and catabolic pathway. So anabolic pathways lead to the formation of more complex molecules from a simpler molecules with the consumption of energy, like protein from amino acids. Catabolic pathway lead to formation of simpler molecules from a complex molecules. Right? So in a nucleic acid, a phosphate moiety links the 3' carbon of one sugar, one nucleotide to the 5' carbon of the sugar of the succeeding nucleotide. So the bond between the phosphate and the hydroxyl group of sugar is an ester bond, and there is one such ester bond on either side and it is called phosphodiester bond. So glycosidic bond between the 2 monosaccharide units, peptide bond between 2 amino acids, and phosphodiester bond between one phosphate group and the sugar moiety in case of nucleic acid. So what are the 2 types of pathways? One is called the anabolic pathway and the second one is called the catabolic pathway. So what is anabolic pathway? Lead to formation of more complex molecules from a simpler molecules with the consumption of energy. For example, proteins that are formed from amino acids. As we know, amino acids are the building blocks of proteins. So what are catabolic pathways? Lead to the formation of simpler molecule from a complex molecule, example glucose and lactic acid. Then moving on to the next very important concept called enzymes. All of us know enzymes are so very important in various physiological processes, and our whole body is like a chemical factory where a series of chemical reactions are taking place. So all these reactions are mediated by enzymes. So enzymes are nothing but called biological catalysts. They are called as biological catalysts. And I think all of us know enzymes are proteinaceous in nature, but all proteins are not enzymes. And the very important role of enzymes is they speeden up a biochemical reaction. Like for example, like morning we have a breakfast. By the time after 2 hours, we feel hungry. So what has happened to the food that we have taken in the morning? It has been digested. So that process of digestion takes place in presence of enzymes. And do you all think the enzymes are always in an activated state? No. So only when their function is needed, they are activated. So the inactive form of enzyme is called zymogen. So zymogen is inactive enzymes. Why the enzymes are found in an inactive state before they start their function? Because if they are always in an activated state, they are going to damage the cell on which they are going to act upon. So that is the reason they are always in an inactive state, and the inactive state of enzymes is called as zymogen, and there are what we call it as activators, which is going to activate the inactive zymogen into active enzymes. For example, like HCl, the hydrochloric acid that is produced by the gastric glands of the stomach is an activator of an enzyme called inactive enzyme called pepsinogen into pepsin, trypsinogen into trypsin. So the enzymes are not always found in an activated state, but only when their function or job is needed, then they are activated. So the inactive form of enzymes is called zymogen. So enzymes play a very great role, and one very important feature of enzyme is enzyme specificity. Enzyme specificity. What do you mean by enzyme specificity? They are specific in their action. That means the substrate on which the enzymes act are specific. So they work in a lock and key system. They work in a lock and key system. Work in a lock and key system. What do you mean by lock and key system? To open a particular lock, we use a particular key. So in a similar way, for a specific substrate, so the substrate might be a carbohydrate, substrate might be a protein, or substrate might be a lipid. So that particular substrates are acted upon that particular enzymes only. For example, carboxylases are the enzymes which act on carbohydrates, proteases are the enzymes which act on proteins, lipases are the enzymes which act on lipids. So lipases don't act on carbohydrates, carboxylases don't act on lipids, proteases don't act on lipids. So that means they are specific in their action, and that property of enzymes is called enzyme specificity, which work in a lock and key system. Just to open a particular lock, do we use any kind of key? No. For a particular lock, there is a particular key to open. In a similar way, for a particular substrate, there is a particular type of enzyme to act upon. And another very important feature of enzyme is the enzymes are neither used up in a chemical reaction nor the structure is altered. You get back the enzymes as it is, and it can be reused again and again. So I think all of us know enzymes, we don't take it in the form of food. They are synthesized by the body itself through the food we eat. So they are also referred to as micronutrients. They are referred to as micronutrients. So you don't require enzymes and proteins in large quantity. Hence, they are also called as micromolecules. Whereas carbohydrates, proteins, lipids, they are required in larger quantity for the body, and they are also called as macromolecules. And even the molecular weight also differs. And enzymes and vitamins are not taken as food, but they are synthesized by the body itself through the food what we eat. So hence the enzymes and vitamins are also called as micronutrients. So now let us understand what are enzymes. So here you can see the beautiful picture of an enzyme. Here you can see the strip. So this enzyme acts as a key. And this acts as a lock, and this acts as a key. So for a particular lock, there is a particular key. So that is called as enzyme specificity. So we can also call it as as I said, we can use the word called enzyme specificity. For a particular substrate, there is a particular enzyme. So this is substrate actually, and this is the enzyme. This is the enzyme. For a particular enzyme, there is a particular substrate. Now let us understand what are the enzymes. A substance produced by a living organism which acts as a catalyst to bring about a specific biochemical reaction is called as an enzyme. How do we define the word called enzyme? A substance produced by a living organism which acts as I said, these are called as what? Biological catalyst. What is a catalyst? That which speeds up the chemical reaction. That which speeds up the chemical reaction is called as a catalyst. So a substance produced by a living organism which acts as a catalyst to bring about a specific biochemical reaction. So they are called, hence they are called as what? Biocatalysts. Then almost all enzymes are proteins, but all proteins are not enzymes. Like for example, we have ribosome, nucleic acids that behave like enzymes, has primary, secondary and tertiary structure. So what are enzymes? A substance produced by a living organism which acts as a catalyst to bring about a specific biochemical reaction. So they are called as what? Biocatalysts. Almost all enzymes are proteins. A ribosome, nucleic acids that behave like enzymes, then has primary, secondary and tertiary structure. So here we can see the active site. So there is a specific site on the chromosomes on which the enzymes fit on the substrate. So active site of an enzyme is a crevice or a pocket into which the substrate fits. So here you can see this is the active site where the enzyme fits. So you can see the crevice. So the crevice is devised in such a way that the substrate and the enzyme will fix together. So active site of an enzyme is a crevice or a pocket into which substrate fits. Enzymes get damaged at high temperature. Yes, very, very important. I think all of us know proteins get denatured. What do you mean by denatured? Their structure gets altered when it is subjected to high temperature. High temperature. So what are enzymes? Enzymes are nothing but made up of proteins. So when proteins are subjected to high temperature, they get denatured. So also the enzymes get damaged at high temperature. So enzymes isolated from thermophilic organisms live under high temperatures are thermostable. So enzymes isolated from thermophilic. What do you mean by thermophilic organ? That which can survive in extreme high temperatures. They are called as thermophiles. So enzymes isolated from thermophilic organisms live under high temperatures. They are called as thermostable. So enzymes accelerate the reactions many folds. As I said, they speed up a chemical reaction. Like as I said, morning we have breakfast. By the time in the afternoon we feel hungry, and after having lunch again by after 2 hours we feel hungry. So there is a series of biochemical reactions taking place which is because of this enzyme. They speed up a biochemical reaction, thereby carrying on all the physiological processes, particularly the chemical reactions that is going to take place inside the body. So enzymes lower the activation energy of reactions. Then the chemicals on which the enzyme acts are called as substrate. So here substrate might be a carbohydrate, as I said, carbohydrate, then proteins, then lipids. So these are all called as what? Substrates. And enzymes like for example carboxylases are the enzymes which act on carbohydrates. Proteases act on proteins. Lipases act on lipids. So enzyme converts substrates into products. So here enzyme plus substrate forms enzyme substrate complex. Enzyme substrate complex, and this will again give back what? Enzymes plus products. See here you can see you get back the enzymes as it is in a chemical reaction. You get back enzymes as well as products. So the chemicals on which the enzyme act are called as substrate. Enzymes convert substrates into products. So active site of an enzyme is a crevice or a pocket into which the substrate fits. So here you can see the substrate, here you can see the crevice, here you can see how it has fit. So here you get back the enzyme and this is the product. And this is called as enzyme plus enzyme substrate complex. This is called enzyme substrate complex, right? So you get back the products and the enzymes. So you get back the enzymes as it is, as well as we get back the product. You can see the color difference. So you get back the products. Enzymes, that is, active site of an enzyme is a crevice or pocket into which the substrate fits. Then enzymes get damaged at high temperature. Enzymes isolated from thermophilic organisms live under high temperature are thermostable. Enzymes accelerate the reactions many of folds. Then enzymes lower the activation energy of reactions. The chemicals on which the enzyme acts are called as substrate. Enzyme converts substrate into products. Hope you have understood all the concepts what I have explained in this particular session of chapter called Biomolecules. So I'll be back with some more interesting concepts of this very interesting chapter called Biomolecules. Till then, goodbye and thank you.